Hello everybody, and thank you for being here. Thank you also to the organizer for giving me the chance to present. Today I will talk about the glycan assignment and quantification in microalgae using in-cell carbon-13 solid-state NMR. So first, why study glycans? As you can see here, compared to other biomolecules such as protein or DNA that are linear polymers, glycans can be found as a linear but also very complex branch polymer. We have about 20 natural monosaccharides, and these monosaccharides can be polymerized as pure polysaccharide, giving usually something associated with energy storage. For example, in plant we have starch, but we also have phytoglycogen and fructosan, while in animal we have mainly glycogen. Polysaccharide and glycans can also be found decorating eukaryotic cells molecules, such as proteins, for example, and this is actually the main post-translational modification happening on threonine, serine, and hydroxyproline. It can also be found on lipids, and usually those lipids are found in the membrane outer leaflet and are associated with cell-cell interaction, communication, and many other mechanisms. It has to be noted that in bacteria we have pretty few glycoproteins, and this can be annoying for heterologous protein expression. Glycan composition in the cell is very finely tuned and regulated by the cell, but it can also be very dynamic in response to a stress in the media, for example. And since it's very regulated in the cell, it's also associated with many pathologies in humans, for example. Then, why study algae? First, algae are found everywhere. They are found in oceans, freshwater, soil, they are just everywhere. Also, it's already extensively studied for different scientific questions, such as photosynthesis, but also heterologous protein expression. As I said, protein and bacteria can have some limitation in terms of glycosylation, while microalgae are true or real eukaryotic organisms, so they have the glycosylation machinery. Algae in general also have a rapid growth and well-known metabolism. That's useful for us for solid-state NMR because it makes the carbon-13 labeling pretty easy. We also have already available a huge bank of mutants, for example, starch and cell wall mutants, that are known to have polysaccharide in them. In general, algae have potential added value such as starch production, biofuels, metal sequestration, and other environmental questions. So first, let's go with the algae Chlamydomonas strontiaceae and the starch I extracted from it. This first part has been published in 2018 about the starch structure and characterization using solid-state NMR. So first, as I said, it's easy to produce your carbon-13 labeled cells. If you want, you can just spin down your cells, put it in solid-state NMR rotor, and make your measurements. What you can do to have a much simpler model first is to break your cells, extract and purify your starch. And this you can hydrate it because water is very important for starch structure. And then you can again make your solid-state NMR experiment. And this is always to have a better understanding of the whole cell architecture. Here, just focusing on starch for this first part. Starch, as you know, is the most abundant energy storage molecule found in plant. It's a carbohydrate made of glucose unit linked together as a linear architecture called the amylose, but it also happens to have a branch structure called the amylopectin. Starch is a very crystalline and very rigid structure found in the cell and can be found into two types of starch, the A-type starch, such as maize starch, and B-type starch, such as the potato starch. We also have, of course, some amorphous part in the starch. To study glycans and here starch, we usually use this experiment called the carbon-13 inadequate. As you can see here, an example of the spectra we get from starch, extracted starch. Why use this experiment? Well, glycans have a very limited chemical shift dispersion. You can have just between 60 and 110 ppm, all your glycans stuck all together. So you need to spread a maximum your chemical shifts. And that's why instead of here on the y-axis having the single quantum, you have the double quantum chemical shifts here, spreading your chemical shift along this axis. As I said, you have differences between two types of starch, A and B type starch, as you can see here in red and blue. And you just have few differences. I zoomed in here. I won't discuss about it today, but it's described in our paper. As you can see, it's pretty easy to make your spin system correlation assignment here. So all the 6 carbon of the glucose unit, This paper I just described, I just mentioned, also was the first one to describe the non-reducing end group of the starch here. Non-reducing end group, it just means it's the end of your polymer. Now we have assigned starch on an isolated sample. We can also make measurement in whole cell. So you just, as I said, spin down your cells and put it in the rotor and make your experiment. This is the kind of spectrum you get with whole cell. So as you can see, we have much more peaks here. And comparing the starchless strain and the wild type strain, we just have few peaks disappearing. And when I copy paste the assignment I just did before, we can just see that we have exactly the same chemical shifts, meaning that our extraction is not destroying starch. That's expected because starch is so rigid and so crystalline. But we still observe some overlap with other glycans in the cell, that's expected too. And we also assign other metabolic byproducts associated with the starch metabolism, such as, again, the non-reducing end groups. This work was the first one to make in-cell starch type identification between A and B type starch using two-dimensional solid state NMR. And just a quick note here, Chlamydomonas inerti has an A-type starch here. So just to wrap up this first part, this work we published was the first one to unambiguously assign the A and B type starch using 2D NMR, but also the non-reducing end group and the amorphous starch. We absolutely need the 2D NMR, so don't forget you need label samples here. We use also this assignment to make in vivo measurements, first to identify what kind of starch type you have, but also to follow starch metabolism. As you can see here on the bottom right, using 2D, you can integrate your peaks and have an idea of the starch intensity quantification you have. And as you can see here, we have different strains, the wild types, the amylopectin rich, the amylose rich, and the starchless strain. And it's described in literature that nitrogen starvation increases the storage of in the microalgae in general. And here, that's exactly what we see. And as you can see, of course, this starchless strain doesn't have any starch, never had any starch. But we also saw a big increase in the lipid bodies content. So now let's go back to whole cell. As you can see here, we have much more peak than just starch here. As you can see here, it's again easy to find because it's pretty intense in the algae. But now I will change from Chlamydomonas inerti to Parachlorella beijerinckii CK5 algae, just to change the system. As you can see here, before it was a 600 MHz spectra, and now it's a 800 MHz spectra acquired at the National MagLab facility, allowing to have a much better resolution. Using this resolution, we took our time to notice and to assign all the assignment we had. I won't go, of course, into detail of all the correlation, but just to give you an idea, you always need to use database to see what is possible in terms of spin system you assign. And for example, here I use the CCMRD database, which is the database developed by Professor Tua Wang at the Louisiana State University. So sometimes for a glycan, it's kind of easy to assign it because you have a very specific, for example, carbon 1 in arabinose, you have a very specific chemical shift. But sometimes not only one carbon chemical shift is important, it's more the pattern of this spin system you just assign. So it's always time consuming. And as you can see here, we have a lot of overlap. So after assignment, we wanted to make quantification, and overlap is very annoying. But if you take your time and make it very carefully, you can have some quantification of your different glycans within the cell. You can take into account the overlap with other, you can't take into account the overlap with other type of glycan, but you can take into account overlapping within, for example, different arabinose. And this is what we get using all these green squares integrals I did. And here you can see the quantification we found after. And as you can see here, we have about 20% loss of glucose intensity compared to mass spectrometry here in black. Just a quick note here, orange is the monosaccharide signal, while red is polysaccharide signal. And the difference is mainly coming from the carbon 1 chemical shift. But so we would try to find where it comes from this loss in intensity. And we found out that comparing the 1D experiment of a direct pulse with different recycling delay of 3 seconds, that's what we use for the inadequate to have something in terms of hour of acquisition, comparing with 30 seconds, where all the T1 will be, all the carbons will be relaxed, then you can see a big loss in the intensity. If you notice here, it's very small, but you can notice that some peaks coming from other polysaccharide are fine. So it means that the most affected polysaccharide in the cell by this short recycling delay is the starch. And it's so huge that it leads to a big loss in the intensity. So as you can see here, we found this correction parameter here to adjust the start intensity. So when you make your quantification, be very careful of your T1 relaxation. So from this quantification, adjusting the intensity just of starch, that's what we get. And as you can see here now, mass spectrometry and solid state NMR match pretty well. But here again, might need high field to have good resolution. Always take care of your T1 relaxation, which is mostly a problem for very crystalline molecules such as starch. And here again, it's not only starch that has been quantified, but also all the other type of glycans within the cell directly. Just a bit of perspective of our current work. So as I said, assignment of glycan can be complicated. So we are working on a statistical analysis of the different chemical shifts available, of the different spin system, to help people to make assignment of the different glycans, not only based on one carbon chemical shift, but on the patterns of spin system. We're also working on glycans from other extract, other natural products, such as cellulosic or glycoprotein-rich cell walls, and also glycolipids, which are important again for cell-cell interaction. Where solid-state NMR is not only quantitative and not only useful for assignment and quantification, of course, it can also be very interesting and used, that's what we are doing currently, for dynamics and hydration comparison and quantifications. And this overall is to build a better and to build new glycan architecture model, and also using NMR, a possibility to make short and long range distances contact. And this, for example, might be used to observe very at an atomic scale the contact between glycans and amino acids. With that, I would like to thank my supervisor, Professor Isabelle Marcotte, and Dr. Dror Warshawski, but also Professor Tuo Wang, where I did a 6 months internship where I learned a lot in terms of glycan assignment and quantification. And I thank also all the people at the National MagLab facility where I did some of the 2D experiment you saw. And thank you for your attention. And if you have any question, it will be later. Thank you.
That's right. Thank you very much. The questions will be later. So we'll have our next speaker, Ahallya. Would you like to share your screen now? Thank you. Would you happen to be still muted, Ahallya? We can't hear you.
Hello, everyone. I'm Ahallya. I'm a second-year PhD student here at IISc. Today, I'll be sharing a B1 calibration method, which we have developed based on offset-dependent mutation experiment using a small molecule. Offset-dependent mutation experiment is very much like CEST, but there will be no chemical exchange or saturation. CEST experiments are usually used to characterize exchange processes with exchange rates in the order 10 to 100 per second. And in the CEST experiment, we start with the Z magnetization of the target nucleus and perturb it using a weak B1 field for a fixed exchange time at different resonance offsets. When we sweep the spectrum with B1 field, when the B1 field is on resonance with a peak, you will have a dip in intensity, which will correspond to the major dip in the CEST profile. And in a limiting case where there is no exchange, the magnetization MZ will simply nutate in a plane perpendicular to the applied B1 field. So when I say offset dependent nutation experiment, this is what I'm referring to. In case there is an exchange and A is an exchange with B, you will get another dip in the CES profile. And from the CES profile, you will be able to extract the exchange rates, chemical shift difference, etc. B1 calibration is very important to get reliable values for the thermodynamic parameters from the CES profile. In fact, we were using CEST, a selective 1D CEST experiment to study solvent exchange rates in sucrose using the 2-bond deuterium isotope effect on carbon. This is the pulse sequence we were using. We use a cross-polarization transfer with Hartmann-Hunt condition for the selectivity. So the HY magnetization is transferred as a CX after the cross-polarization, followed by a 90-degree pulse to bring the carbon magnetization to Z-axis during the exchange period. And then you detect the intensity of the CZ magnetization after the exchange period by transferring it back to proton. This is one of like a samples of selective 1D proton spectrum, which we acquired using the pulse sequence. This is a peak for the anomeric GC1 in the sucrose. We use the same, all the data which I show of the offset dependent nutation experiment, we acquired using the same pulse sequence. In that, since the magnetization is just mutating about the B1 period during the time delay, we'll refer to this period as mutation period. So when we were studying the solvent exchange rates in sucrose, we observed mutations. We actually observed modulations in the CES profiles where we did not expect it. And later we realized that these modulations were actually these mutation profiles of the magnetization as a function of the offset. In fact, if you solve the block equations for a limiting condition without exchange and no relaxation, you will see that the MZ is indeed a function of sin square. And at very low B1s, the mutation profile matches very well with the 1 minus sin squared function. And the P here is a function of both the intensity of the B1 field applied and where on the spectrum you are. So these mutation profiles must be very sensitive to, or at least they are very dependent on the intensity of the B1 field and the offset. To see how sensitive these modulations are actually to the B1 field and offset, we ran some simulations and it turns out that they are indeed very sensitive to the B1 field. The simulations here differ in B1 of say 0.01%. And you can see that even if they are the same pattern, the intensity does vary quite a bit, which means we should be able to fit them to the block equations and extract a B1 value. I would also like to point out how sensitive these mutation profiles are to the B1 inhomogeneity, especially at higher B1 fields. At 50 Hertz, it's almost surprising that the mutation profiles are so different. As you go to lower B1 fields, the mutation profiles do not change as much with inhomogeneity, which is disappointing because we probably wouldn't be able to fit the mutation profile to estimate the B1 inhomogeneity for weak B1 fields. So as the simulations were promising that we will be able to obtain or get a good estimate of the B1 values, we went ahead and fit our mutation data to the block equations. So these are the mutation profiles of the GC1 carbon of sucrose, that is the anomeric carbon, which doesn't have an exchange. It doesn't have an exchange because there is no 2-bond deuterium isotope effect. So as you can see, we have pretty good fits. We also do the fitting by minimizing the chi-square function, the energy function, and we fit B1 inhomogeneity, relaxation rates, and the chemical shift off resonance. When I say chemical shift off resonance, it means, so we are measuring one peak at a time. And this will be how off your carrier is from the actual chemical shift of the peak. So visually, it would be how off-center the center of the nutation profile will be from 0. So going on to check the robustness of our fitting, we ran reduced chi-square surfaces, and they converge very well. To obtain the errors in the fits we have obtained, we ran bootstrap error analysis, and we obtained an error of 0.01 hertz for most of the B1 fields. Now to validate the B1 fits which we have generated, we wanted to compare it with the existing method. So on-resonance mutation experiment is something which is standard and used for B1 calibration during CEST. In this experiment, you apply a B1 field on resonance to the peak and let the MZ magnetization mutate and measure it as a function of time. So in this case, your offset is fixed and you vary the nutation time period, which is complementary to what we do in our calibration method, where we sweep the B1 across the offset, keeping the nutation period constant. Now the MZ would mutate in a plane perpendicular to the B1 field with the, as a function of the B1. So if you measure MZ with time, you will get it as a cost function of ω1. You can Fourier transform that and get the spectrum, which will read out the B1 field intensity. So the indirect dimension here will be the power of the B1 radio frequency field. Comparing the values which we obtained using the existing on-resonance mutation method and the one which we estimated using our offset dependent mutation method, you can see that the correlation is pretty good. So we wanted to, what you're proposing is that we can use a small molecule like sucrose to calibrate your B1 fields for experiments like CEST. For proteins, we have a set of CEST experiments for different regions in the spectrum, C alpha CEST, the carbonyl CEST, and yeah. So, but it is questionable if we can calibrate all of these regions in the spectrum using just sucrose. To evaluate if it is possible, we created a sample of small molecules, benzaldehyde, sucrose, and alpha ketobutyrate, so that we have a range of offsets on which we can calibrate. So here you would be able to get an estimate of the B1, which can be used for the carbonyl CEST using the benzaldehyde chemical shifts. You can run the methyl CEST using the alpha ketobutyrate. This is in case the B1 fitted deviation is large when you use these offsets. So here is a histogram of the B1 values which we calibrated on all of these different peaks. What we observed is that there is a variation of 1.3% in the average variation of 1.3% in the B1 calibration using sucrose and benzaldehyde and alpha ketobutyrate, which are on two other ends of the spectrum. We wanted to extend this method to 15N calibration too, and tryptophan seemed like one of the good molecules to calibrate proteins. Shown here are two mutation profiles of tryptophan at different B1 fields. Just like sucrose, the fits were reliable. We had to produce correct convergence and error estimation. Now, we have two ways to use a small molecule as a standard for B1 calibration. We can either use this as an internal standard with the protein or have it as an external standard. Ideally, it wouldn't be desirable to have it as an internal standard. Anyways, so we compared the B1 calibrated using our method and the one using the on-resonance mutation for the tryptophan ubiquitin sample. So here the on-resonance mutation experiment was done on an isolated peak on ubiquitin, and the offset-dependent mutation calibration using our method was done on the tryptophan. You can see that the correlation between both of these values are pretty good. But having an internal standard isn't ideal. So we wanted to check if having it as an external standard would work. And the histograms here shows that they have pretty good values, it matches each other. In fact, the error is 0.4% for tryptophan internal to tryptophan external B1 calibration, which means we can indeed have tryptophan as an external standard. But the catch is you should acquire the mutation profile for the external standard right after the protein without tuning, matching, or calibrating the pulse sequences. If you tune, match, and calibrate, we see a larger deviation in the fitted B1 values. So thus we have validated that the proposed method is working as good as the existing on-resonance mutation. Now the question if there are any advantages to this method over the on-resonance mutation experiment. In the on-resonance mutation experiment, your B1 field has to be exactly on resonance to the peak. If there is a variation, that is, if it's slightly off resonance by, say, even 1 or 2 hertz, there will be a chemical shift vector, and your MZ magnetization will end up mutating about the B1 effector, which wouldn't be the actual applied B1 field. Now, this is not as big a problem if you're calibrating higher B1 fields like 50 hertz, because it's very unlikely that you will be off resonance from the peak by 50 hertz. But if you're calibrating something as small as 1 hertz, 2 hertz, or anything less than 5 hertz, it would be a big deviation which you will get. This is a plot which we of the B1 calibrated values using the on-resonance mutation and the our method of offset dependent mutation. As you can see, when the chemical shift off-resonance increases, the B1 calibrated values of mutation definitely increases as a function of this, whereas the value which we have fit using the modulations in the CES profile stays constant. This is because in our fitting, we also fit the chemical shift off resonance, and the off resonance effect wouldn't have any effect on B1 calibration. It's also nice that we can have an estimate of B1 inhomogeneity using this method. Although at lower B1 fields, we don't think that we'll be able to get a very good estimate. But for B1s above 5 hertz, we have gotten good estimates of B1 inhomogeneity. So yeah, what we are proposing is a method for B1 calibration using the modulations in the mutation profile, which using a small molecule. Because if it's a big molecule, the relaxation rates will be too high and the modulation wouldn't be as prominent. Okay. Yeah, this is everything we have right now on the offset dependent B1 calibration method. We calibrate our CEST experiments using this method, and it has been pretty effective. I would love to hear your thoughts on the method, and we are on the lookout for appropriate acronyms for the method. So if you have anything in your mind, please do share. So Clarice and I worked on this project together. She's always high on her spirits, like in the picture. Ashok is our PhD advisor who has guided us every step of the way. Our lab is a new lab here at IISc, and I want to thank every lab member has contributed to the project in certain way. I want to thank especially Ajit, Bodhi, Madhu, and Vaishali for the help they have provided. That is all I have for now. Thank you for listening.